Yep.
Hi, everyone. Thank you for joining us this evening. I'm Stephanie. If you don't know who I am the chapter president for the Broward chapter of Florida Native Plant Society. We're really excited for y'all to be here. We have a lot of people that are logged in tonight, a bigger crowd on Zoom than we typically have, so this is great. We also have an audience here at Secret Woods, so we're really happy you could join us. Just one housekeeping I wanted to mention is that we have our field trip this Saturday from 10 a.m. to noon at Pam's Garden in Palm Beach. If you're interested in attending, please email the email address that was in the newsletter or the email address that's on our calendar, just the RSVP. Right now we have a small group that says that they're attending, so we can definitely have more attendees if you're interested. So Susan Lerner, who's the director of Pam's Garden, hopefully will be there, and she can tour us around. She also was the previous president of Palm Beach County chapter of Florida Native Plant Society. So a lot of Florida native plant enthusiasts and everything, so it'll be really great. Hopefully you can join us. And then yes, so this evening we have George Debs, and I'm going to let him introduce himself. So
Great, thank you. Thanks everyone for being here. Anyone that's online, if you have, can you confirm that you can hear us fine?
Yes, I can hear you.
Yep,
Yep.
I can hear you.
Great, and if everyone can just mute their microphone, just so that we don't have any hiccups during the call. That would be great. All good?
awesome. I think so. I'll just sit here in case anyone comes in late to admit.
Great. Just a moment, I'll run this. Okay. So thanks for being here. It's great to finally be on this side of the table. I've watched so many from the other side. My name is George Debs. I'm a designer at Cadence. I work with Stephanie. And I am a certified permaculture practitioner. I got to travel to Australia back in 2015, where I volunteered under Jeff Lawton. And most recently, I've been studying with Dr. Elaine Ingham, who some of you might have heard of, might have not heard of, and I will be talking about her in a moment. And I've been training to be a certified soil food web lab technician. So I'm able to take soil samples from the field and test them for biology. And we're gonna get a better understanding as to what all of that encompasses and why that's so important. So this is an introductory presentation. This is a really vast topic. There's so many things that we can cover. We can have so many presentations about it for hours. So I'm gonna keep it as succinct as possible. I want to cover as much ground as possible so that you guys get a better understanding about what the soil food web is, and hopefully it can really open up your minds and eyes to the world around you and see things in a different way, and how you interact with your landscape as well. So let's jump right in. Hopefully this works. Oh, there we go. So general outline, I'll just be quickly going through how nature functions versus how conventional agriculture seems to be functioning currently. Then we'll jump into what the soil food web is, why it's so important. And then at the end, we'll talk about how we can all start to take deliberate steps within our own landscapes and gardens to reincorporate and reintroduce the soil food web. And then we'll have a Q&A session. So I want everyone to take a moment and think about a forest. This image here is a cypress swamp that Stephanie was nice enough to share with me. And think about how there's so much life in the forest. There's so much growth, so much biomass that comes up, but there's no tilling. There's no copious amount of fertilizers that are being added in seasonally. There's no spraying, but the system continues to grow. It continues to complexify. So in contrast, think of conventional agriculture. There's usually very high human inputs. We bring diversity all the way down. That usually results in soil erosion. It results in pollution and decreased yields over time. You know, in nature, technically, nature doesn't need it needs zero human input. Now, indigenous cultures have usually a very positive effect and synergy with these ecosystems, and they tend to benefit them and progress them. But technically speaking, nature doesn't need human input, but it still is able to support complex life. It's still able to self-replicate perpetually and build soil and filter toxins. So how is this possible? How is it that nature is able to have this synergy with all these different organisms and continue to complexify with very minimal, seemingly minimal input. And we're finding, the research is showing and has been showing, that it's really the soil food web that drives a lot of this. So we're going to jump into what that is. But before that, I do want to mention Dr. Elaine Ingham, who's my professor. She is a world-renowned soil microbiologist. She's a pioneer in soil biology research. She's been in the field for over 40 years, doing countless studies on what the soil food web is, and really has brought it to the forefront of the latest, in agriculture and ecological restoration. And in 1996, she started the Soil Food Web School to really help bring this information out into the public and really start to educate growers and professionals on this. So kudos to her for bringing us all on this trip. So jumping right in, this is probably the most important diagram in everything I've studied. It encompasses everything. And I could give the entire presentation on this diagram, but I will spare you that. And it's called the soil food web. Some people call it the soil biome. And to keep it succinct, essentially, it is all these different organisms, billions and billions of organisms that interact with each other. They create a balanced ecosystem within the soil and also on plant surfaces. And they vary in sizes. Usually it's organized via trophic level, which is like a hierarchy where each level is larger usually and predates on the smaller levels. And they all kind of have these different exchanges, and we call it a web because if you remove one piece from the entire system, it really has an effect on the balance of the entire system, and things can get really out of balance. And plants fit into this too. So we'll jump into how all this works. We're going to admit some more people. Great. So how do we study the soil food web? Most of these organisms are microscopic. We can't actually see them with our naked eye. So we have to use tools such as a microscope. So this is a typical view when you look through a microscope. This is magnified 440 times. It's a very small piece of moss. But when we're talking soil food web, we want to look even larger than that. This is 100 times. This is a leaf of a moss leaf. You can even see the individual cells. But when we're really wanting to quantify and understand what's happening in the soil, we want to magnify up to 400 times. And there's a lot going on in this slide. There's aggregates. I can see humic acids and forming and fungal strands. If this was a video, you'd see things zooming around, and we'll see some footage of that later on. So this is a very typical thing. And this is a good sort of sample that we want to look at. So taking a step back, looking back at that diagram, so what is the soil food web? Why is it here? How does it even function? So studies are showing that it varies from plant to plant and species to species, but many plants can release up to 80% of their exudates into the soil. So what are exudates? We all know that plants photosynthesize. So they create photosynthates, things like carbohydrates, sugars, proteins. They'll produce these within their bodies. And they're finding that they're actually releasing up to 80% into the rhizosphere, which is the root zone. So why would a plant waste 80% of what it's making? It's not actually wasting it. There's billions of years of coevolution happening here. They actually have symbiotic exchanges with microbes within the soil. So this is essentially the driver for the entire soil food web. And we're going to delve into what this all means for our plants. So there is a reliance from either side within the entire system. So what does the soil food web do? We're going to jump into all these different benefits that the soil food web has. And these are some of them, and we'll delve into them. Things like suppressing pests and diseases. They'll help retain nutrients within the soil. They'll manage nutrient cycling between the plants and the soil. They'll build soil structure. They'll even decompose toxins and pollutants as they pass through the system. And they can drive succession, which is really, really cool and important, especially when it comes to our gardens. And we'll delve into that. So how does the soil food web suppress pests and diseases? So the image on the left here, you can see that is a plant surface, a leaf surface under electron microscope. So this is, you can't see this with a naked eye, but essentially the plant surface is covered with organisms. So there's bacteria here, there's fungal strands, There's different predators that move along the plant's surface. So if you're out in the wild and a mold spore lands on the plant or a pest lands on the plant, it doesn't actually land on the plant's surface. It lands on this biome that is living on the plant. So these organisms basically outcompete anything that lands on the plant. They inhibit it, they consume it before it can even populate. The image in the center here is denuded leaf surface. It's something you typically find in conventional agriculture. We've eradicated all the organisms, and anything that lands will basically land on the plant surface, have no competition, and can just spread and create a massive population. And here you can see bacteria that's created just a singular population. And this is what we see with the naked eye as an infestation or a disease that happens, something like powdery mildew or sooty mold on a plant surface. So the soil food web helps mitigate that. It helps prevent this from happening. This is a really, really interesting slide, and nutrient cycling is essentially these symbiotic exchanges that happen with plants. What the organisms are doing is they are mining for minerals within the soil and providing them to the plants in exchange for these carbohydrates and proteins and sugars. So think of a little bit of carbs, a little bit of protein, some sugar. I mean, that's a recipe for cakes and cookies, essentially. So the plants are releasing all these different sorts of cakes and cookies all along their root surface, attracting different sorts of organisms to those sites. And then they're not just giving it up for free, they're asking in exchange for different minerals from the soil.
You're going to keep going, babe.
And this is something that's pH specific. So bacteria and fungi function at different pHs along the plant's surface. So this diagram here on the right, which some of us have seen, talks about how different elements within the soil are only available to plants within certain pHs. So something like iron, for example, typically needs something that's more acidic. So why is it, think back at that forest, why is it that some of our natural forests in Florida, where our soil is mostly alkaline because it's based on lime rock, why is it that these plants don't have an iron deficiency? It's because they have an association with fungi within the soil that actually create micro pHs along the plant, along the root surface and within the soil. And they're able to pull with their acids these iron elements from the crystalline structures in the sand and the silt and the clay that's inside the ground. So it doesn't really matter what your overall pH is if you have a balanced soil food web. Your plant is basically telling this organism, I want 5 grams of molybdenum or 2 grams of boron. And these organisms will go out, create the specific pHs and bring those to the plant. So this is really, really important, and it's really turning a lot of agriculture practices on their heads. Another thing is predation. So, I mean, we like to call it the poop loop, and I'll explain why. Essentially, not all the organisms exchange with plants directly, but a lot of bacteria and fungi still mine for these minerals within the soil as a food source. So when they mine for it, they lock it up within their bodies, and it's not available to the plants. So this is where a balanced soil food web comes in play. You have predators that are the higher trophic levels that eat these bacteria and fungi and essentially poop it out in a bioavailable form at the root surface. So this is why we call it the poop loop. And another thing is humic acid production. So humic acid essentially is, if we look at this diagram here in the center, this nitrogen cycle, it talks about how nitrogen moves within the soil. And a lot of times it actually cycles within a couple steps over and over and over again. Fungi will mine for nitrogen, they'll lock it in their bodies, a predator will come along, eat the fungi, it'll poop it out, the plant might get it, might not get it. Another fungi comes along, decomposes that poop, and reprocesses it within its body, and the cycle goes on and on and on. And over time, what happens is the molecules get more and more complex over time. They get bound up in more complex carbon branches within the fungi. And what this does, it actually locks up things like carbon from the atmosphere. And it's we call these humic acids. And humic acids are very long-lasting molecules within the soil because they're so complex, they don't break down very easily. So this is having major implications for carbon sequestration in the climate crisis, because if we restore the soil food web, we're able to draw a lot of carbon back from the atmosphere and lock it in the soil in a very, very stable form. These two images here on the bottom left. So the one on the far left is a root feeding nematode. And some of you might have heard of nematodes and them being root not feeding nematodes. They're not beneficial to our plants. And we tend to do things like solarization to kill the nematodes. The one on the right is much larger than it. And it's not eating cakes and cookies in this case, it's eating the root feeding nematode. So what we're doing with solarization often is we're actually killing even the predators that would keep these root feeding nematodes in check and keeping them from being a problem. So moving on, another thing that the soil food web does via all these interactions is build soil structure. So these aren't cookies. I know there's a lot of cakes and cookies going on, but these are actually soil aggregates. And bacteria create, via their metabolism, they create things that called alkaline glues. We call them alkaline glues. And fungi create different acids. So the bacteria are able to glue themselves to micro aggregates within the soil. And this is basically kind of a survival tactic for them. They're able to bind all these different particles together to create little niches and safe environments for themselves. These micro aggregates are then taken by fungi and bound together in even larger aggregates. What this does is it creates this kind of crumbly texture to the soil that you guys might have seen if you've ever dug in really healthy soil. It has that crumbly texture that allows oxygen and air to pass through. So we all know that our plants need water to pass through their roots, but they also need oxygen just as equally. So this is really important to make sure that the soil isn't compacted. So I have the number here, 150 PSI. Most plant roots can't penetrate anything above 150 PSI. Some plants with tap roots can penetrate up to 300, but oftentimes in urban contexts, we're finding that they're going well beyond 300 PSI. So plants aren't even able to penetrate that deeply. They just don't have the physical capacity to. But the soil food web, because it's working on such a microscopic level, is able to, over time, break up these compaction layers and build that structure again. And what this does ultimately is allows oxygen to pass through, allows water to pass through much more deeply, and it creates a larger sponge within the soil. So our plants have access, because they have deeper roots, to more water over the season. You end up watering this. Another thing here is, like I said, create aerobic conditions. So aerobic means there's a high level of oxygen above a certain amount. So these organisms actually need a certain amount of oxygen, very similar to our plants. But what happens is, if you get compaction, if you don't have these organisms, you lack oxygen within the soil. And there are organisms that live within these low oxygen conditions. And we call them anaerobic organisms. And they can be fungi, they can be different sorts of bacteria, different types of ciliates. And the thing with these is they are actually detrimental to the plants, because within a low oxygen condition, the sort of chemical reactions that happen create things like alcohols and different acids that are actually toxic to our plant roots. So if you've probably seen root rot, that usually what happens when you have waterlogged systems where there's no oxygen in the system. And what happens is you actually get these different acids and alcohols that are produced. And alcohol is great if you're harvesting it from yeast to make a drink, but pour that on your plants, you're going to burn and disintegrate the plant cells. So that's why having a good soil food web is so important, because it alleviates compaction, it allows oxygen to pass through, and it doesn't allow these organisms, these anaerobic organisms, to thrive and create these conditions that aren't conducive to plants. So driving succession, and this one is really important when it comes to our gardens specifically, and how we're wanting to design with native plants. And there's a lot of biochemistry here, and I'm going to keep it as succinct as possible, but essentially different sorts of plants need different forms of nitrogen to function. Nitrogen usually takes two forms within the soil, typically when it's taken up by plants. It's nitrates and ammonia. Nitrates are things like weeds, things like annuals, short-lived plants usually thrive on a lot of nitrates. And more woody plants, more complex plants that have more complex metabolisms to create these lignins, to create their strong structures within their bodies, need more ammonium. It just depends on how the chemistry and how the metabolism and chemical reactions happen. And what dictates this is the organisms. They are able to process the nitrogen and have it in a certain form. They can have it as nitrate or ammonium. So succession, most of you have probably heard of succession. It talks about the evolution of ecosystems and communities, natural communities over time. So you can start with something like bare rock, and it turns into a weedy patch, and it turns into eventually a forest or an evergreen or deciduous forest. And this is really what we're finding is it's dictated by the biology, not by the plants. So you can start with bare rock where there's very little organic matter, which is usually the fuel for the entire system. And you have just a little bit of bacteria. In this case, you'll have like 10 micrograms per gram of soil. And moving on, as those bacteria start to mine for minerals, they'll form some organic matter via their own dead bodies, and that will allow more different sorts of bacteria to grow, eventually mosses, and then eventually weeds. And as those plants start to succeed, their organic matter is taken up by different fungi. And as the system progresses, and this can take thousands of years to happen, eventually what happens is you have an exponential growth of fungi through the system, and fungi are able to create the ammonium that allows more complex organisms to survive and to grow and to create their different molecules that they need. And what happens is, well, you might be thinking, well, okay, the Earth is billions of years old. Why isn't everything a forest at this point? I mean, it's been going on for this long. And that has to do with disturbance. So disturbance inhibits succession. And disturbance can be things like a flood. It can be things like wildfire, which we're all very familiar with here in Florida. And it can be things like paving over an area. It can be compaction. And all of these things that inhibit the organisms from surviving will drive succession back. And in a case of a wildfire, we call this secondary succession because we already have a system in place. There's still a bank of organic matter within the soil. These organisms go dormant during a fire, or they run away. Nematodes are able to migrate deeper into the soil surface. And the system is able to bounce back a lot quicker because it already has the reserves there to build the system back up. And this is a different form of the same diagram. And the thing to remember here is the same rules apply to our garden. It really doesn't matter what the scale is. The rules apply. We're talking about microscopic organisms. They're everywhere. So if you want to grow a weedy annual patch, and you just need a lot of bacteria. But if you're trying to grow a lot of woody plants, if you're trying to restore a pine woodland, but all you're getting are weeds, that tells you something very clear as to what's going on in the soil. If there's a lot of weeds, but you're trying to grow pine trees, those weeds are interacting with a lot of bacteria that aren't able to produce the nitrates, the ammonium that pine trees need. They're only producing nitrates. So this really helps and answers a lot of questions. We're able to say, oh, I have a lot of weeds, which means I have a lot of bacteria. So I need to introduce more fungi into the system so that I can create the ammonium that these trees are going to need if they're going to succeed for a long period of time. And there's a lot of complexity here. One thing that happens is mycorrhizal colonization, which is really interesting. And some of you might have seen this image before. It's a pine seedling. And this network that you're seeing is actually a fungal network. These fungi are able to create direct symbiotic associations with these seedlings to get all the minerals that they need. But if you have a system that is too early in succession, things like actinobacterial will suppress mycorrhizae from taking hold. So understanding where we sit here really helps us understand what the potential is, what steps we can take to move a system in a direction that we want it to move. So what can we do to restore the soil food web? So I love this image here on the left. It shows, it's a petri dish that shows the mycelial network. And this isn't something that you can typically see with that you see in a garden because this is happening underground. But what's really cool is you see how the mushrooms form. They're very, very small. And when you look under the microscope, you can even see these shapes in a microscopic level. And this large mushroom that's forming here is thousands of times bigger. This is a very complex organism. It's able to do this. So if you see a mushroom like this on the surface, it tells you there is a lot going on under the ground that you don't see. So step one is observing your land and understanding what is going on. Like I said, going back to succession, knowing where you are on that successional stage and knowing if you have mushrooms happening, that means you're probably later in the successional stage, and that's a very good thing. Other things we can do are decompact and protect the soil, and we're going to get into these and how we can actually practice these. So decompacting and protecting the soil is really, really important, especially in urban environments where a lot of times when we build homes, we have to decompact this, we have to compact the soil for foundations, or you have a lot of vehicles that pass over areas. And there's a lot of history. I mean, you've purchased the land and you don't know what's happened in the past. So decompacting and taking approaches like no-till gardening are really important because, like we talked about, if you compact the soil above 150 psi, your plants are not going to be able to penetrate that. You're not going to be able to have a deep system of organisms that are allowing oxygen and water to pass through deep into the soil. So decompacting is really, really important. And you can do it as simply with a broad fork or a garden fork, and just decompacting and not stepping over the area and allowing more rain and more oxygen to get through the system to encourage these organisms to break up the compaction layers for us. Covering the soil is really, really important too, because if you have soil that's exposed, a lot of things happen. First of all, the sun can denature a lot of organisms. That's why a lot of them live underground. They're not able to cope with the UV rays. A lot of erosion happens. So you lose a lot of organic matter to rain, to floods, to wind that happens over time. So if you cover that system, the best thing you can cover with is plants. So you can start with mulch, and that's a great start because mulch is organic matter. It feeds these organisms, it protects the soil, it absorbs a lot of pressure. But ultimately you want to cover the soil with plants because the plants are living matter that are creating these exudates. So they're able to, these people. Yeah. They're able to, sorry.
No, you're fine.
Technical.
How do I do this?
Just exit out.
Just exit.
Yeah. Thanks. So these plants are creating exudates. So they're actively perpetuating the system, much like a forest does. They're able to constantly feed the bacteria and the fungi over a long period of time, and not just them, also the predators. So rain causes a lot of compaction, and you might think, well, one raindrop isn't going to cause that much pressure on a soil, but think of the millions and millions, if not trillions of raindrops that fall on exposed soil. That actually exerts a lot of pressure on the soil, and it can create compaction layers deeper in the soil that we can. So covering the soil, making sure that that rain doesn't create those compaction layers to begin with is the most important thing. And then also system-oriented synthetic fertilizers and sprays. So we've learned that if you have a balanced soil food web, you don't need to fertilize, or you don't need to add synthetic fertilizers. What those, they actually eradicate the organisms. They're not able to cope with that sort of condition that those sprays create. Even going back with pests and diseases on plant surfaces, your plant surfaces are covered with these organisms. So if you eradicate them, you don't have any protection. So as long as you have the soil food web that is able to also, so one thing that happens is, going back to the soil food web diagram, the larger organisms are kind of taxi cabs. They're able to carry these organisms from within the soil and move them onto your plant surfaces. So this is why not spraying different larger insects is so important, because they're actually able to move these organisms, these more microscopic organisms, longer distances than they're able to traverse on their own. So a couple things that we can do to quickly bring or encourage these organisms in gardens. So sheet mulching is a great practice. Mulching, some of you might have heard of it. It's also called lasagna gardening. And it's a great way to replace lawn. I know a lot of us are working towards replacing our traditional lawns with native gardens. And this is a great way of replacing the lawn without removing the lawn. What happens is when you remove lawn, and there are moments where removing lawn can be appropriate, but what happens is you're actually exposing the soil. And so you're exposing all these organisms and you're losing a lot of the biology. So what you can do is actually cover the soil with some sort of occlusion layer like cardboard or brown paper or newspapers, and wet the area and actually suppress and suffocate the existing plants like a lawn, and or if you have weeds, and then covering that with something as simple as mulch. mulch or branch mulch. And there are different ways that people do this. You can add manure in, you can add compost in, but the simplest way to do it is just covering it with cardboard and wood chips. I'm just gonna take a moment. Can we have everyone mute their microphones? It's a little loud on this side. I appreciate that. Thank you. This is an example here, and little photos are a project that I did when I lived in Australia. It was a completely dead forage where I lived. It actually was sprayed with Roundup continually for several years. And I was surprised that there were no weeds that would even grow. So that tells us there was probably hardly any biology going on. So what I did was, the soil was so compacted, I couldn't even dig into the soil. I couldn't even put a garden fork in it. It was absolute concrete. So what I did was I got local horse manure, I laid it down, I watered it as much as I could, even though it would puddle because the water wouldn't even penetrate. I covered it with cardboard that I got for free. And then I covered it with wood chips that I got for free. And it took 6 months to go from the first photo. Here,
Sorry.
I got it. To go from the first photo to the second photo. And this is from adding all that organic matter. I introduced biology via the manure, via the wood chips that were fresh, that came from a different system. I brought them in, and over 6 months, they broke up those compaction layers. So, I mean, this is really a testament. And I've seen so many vegetable gardens, even here in Florida, that are made this way. It's a great, great way to do it. And it's way easier than breaking your back, lifting lawn and getting rid of that. So another really, really important way to inoculate our systems with biology is compost. So at the school, at Soil Food Web School, we talk about creating a biocomplete compost. It's not just a matter of inoculating your soil with bacteria and fungi. You need the predators. You need all the different parts of the system, flagellates and different sorts of protozoa and amoebae that eat the fungi, that make those nutrients available to plants. Because like we said, many organisms don't have those direct interactions with plants. You need the predators in place, which means you need the nematodes, you need the earthworms, you need all the different trophic levels to keep everything in balance. And that ends up leading to larger animals like birds, burrowing animals, ultimately humans. We're all within that system. So there are 3 general ways we make compost. There's thermal compost and vermicompost and static compost. Thermal compost is what we typically like to use because it's a certain size, and it has to be a certain size to reach a certain temperature. So we want to reach certain temperatures to, number one, kill weed seeds, because we're usually collecting materials from the landscape and there's weeds in them. And we don't want to necessarily inoculate all these areas with more weed seeds necessarily. We want to plant our own plants. And then there's specific recipes that we need to follow to reach these specific temperatures. And the important thing here is that it doesn't go anaerobic as well, because if a temperature goes up too high for too long, a lot of the bacteria will use up all the oxygen and it'll go anaerobic, so it'll actually turn on us. So it's very specific. There's certain rules that we have to follow, and what you want your compost to look like. If you're looking for compost to purchase or to add, or if you're making compost, you want it to look like the photo on the right. It should be a 70% chocolate brown color. So go get a 70% dark chocolate bar and compare it to your compost. And the reason I say that is because if your compost is black, and oftentimes we see this, that actually means that the compost has gone anaerobic at some point. Even though it might have cooled down now and it doesn't necessarily smell, it actually has gone anaerobic. And what happens is, what the ammonia that I mentioned, it's actually nitrogen that turns into a gas. So all the work that we're putting into adding all this nitrogen, which is so important for our plants, it actually, in an anaerobic condition, it turns into a gas, into ammonia, and it leaves the system. So your compost isn't a fertilizer anymore. It's kind of dead. It's just maybe organic matter. And when it gases off, it actually leaves a black residue. So when you see black compost, it means it's gone anaerobic at some point. And it's not the end of the world. Typically, when that happens, we just recompost it correctly. And then we reincorporate that organic matter into a thermal compost. We use things like thermometers like this very large thermometer to measure the temperature within a pile and make sure everything's going as planned. And one way we like to cast compost is physically, we like to add compost to the soil. That's a great way of doing it, and then covering it with mulch right away to protect those organisms. But we also create extracts. So we add the compost to water, and we are able to brew it even. And what that does is we're able to multiply, say, 20 gallons of compost and cover hectares of land with all this biology. And then you're also able to apply it to your plant surfaces. Because like we said, this isn't just, it starts in the soil, but it also migrates onto your plant surfaces to protect those plant surfaces. And extracts and teas are a great way to do that. And the most important thing here is aerobic brewing or aerobic extracts. You don't want it sitting for days. You want it to be aerobically. We usually use pumps to pump air through it, and we always are taking samples and looking under the microscope and making sure nothing's going anaerobic, because we can see all the different markers for that. And I mean, this whole slide can be a whole weekend of demonstrations where, you know, this can be exemplified and done. Vermicompost is another great way. Typically, it needs a higher moisture content because worms need, 70% or higher to lay their eggs, which can be tricky because the higher your water content, the more it can become waterlogged and you won't get oxygen in the system. So, but vermicompost is a great way because essentially what earthworms are, sorry, I didn't say what vermicompost is. Vermicompost is earthworm compost. So essentially it's organic matter that earthworms consume, and then what they produce, the fecal matter, is a great compost. And the reason it is great is because essentially worms are tubes full of a very vast diversity of organisms, so, and bacteria specifically. So pathogens, they'll eat pathogens, anything that will fit in their mouth, basically, they'll eat it. And if you have pathogens that they'll consume, those pathogens are outcompeted and inhibited and consumed by all the organisms within the worm. And then what comes out is something that's very pure and very clean and doesn't have these pathogens. So it's a great way. Typically, fungi don't get processed that well because the worms will eat the fungi and then they come out the other end as organic matter. But we're able to inoculate fungi back into those systems. And if you're able to have vermicompost, that's a great way to introduce biology. Static compost is another easier way to make compost. It's less technical. It just takes more time. So usually static compost piles need a year to mature, because what happens in a static compost pile is you're putting all these materials together and you're not turning it, you're not moving it. And I forgot to mention, with thermal compost, we're making the pile, we're testing the temperatures, and then as soon as it reaches a certain temperature, we need to cool it down. So we open up the pile, we mix all the materials together, and we cool that pile back down, and then we allow it to rise back up in temperature a certain number of times. With static compost, you don't do that. You just put the materials together and it will go anaerobic. The center will go anaerobic. But what you're doing is you're waiting a longer period of time. So the outside of the pile will stay aerobic. You have those organisms that have access to more oxygen, and you're hoping that within a year's time, and if you add worms into the system too, that they will slowly eat away at that anaerobic system in the center and make it aerobic. So it takes more time, but if you have that time and you don't want compost very quickly, that's a great way to do it. So that concludes my presentation. I hope that brought some information that enlightened some of you and brought some new things to your mind that you weren't aware of. We'll open it up to Q&A. These are some common questions. If no one has questions, these are some common questions I get, which we can dive into and answer.
And if you're virtually, can you put your questions in the chat and we'll field the questions in person here if anyone has them, and then we'll move to the chat to answer some questions.
Yeah. So any questions? Yeah.
Sometimes when I'm mowing my yard in the backyard, I let my weeds grow about 2 or 3 feet during the summertime because I'm lazy. Then I started cutting with weed whacker, and I found this little bulb, fungi bulb, and I opened it up. It's kind of like dusty inside.
Yeah.
Now, what are those? All healthy? Can I just put it back in the soil? Or whatever?
Yeah, so typically when you see fungi like that, like saprophytic fungi, something like this, or even, a puffball that has like has powder inside it, those are typically, they could be I'm not a mycologist, so there are thousands of fungi, and it's a great thing to get some resources to identify exactly what fungi you have, but that's usually a good thing. It usually means there's definitely decomposition happening. So they're in the soil, they could be having interactions with all these different plants, but the worst thing they could be doing is decomposing material that's sitting there. So typically fungi are a great thing. It's always a good thing to, join an online group or get a book that has, some of your local fungi and get familiar with what you have so you understand, because some of them might be toxic, might not be toxic to us as humans. So, we have to keep that in mind too. But if you see fungi, that's a sign. It's better than not seeing fungi, I'll say that. It means that you have some sort of diversity happening. Yeah. Yeah. Yeah.
You are seeing a whole bunch of. What is it that you're describing as weeds?
So this is a big question, right? So weeds are essentially, the way I define weeds, the way we typically define weeds is they're short-lived, typically annual plants that complete their life cycle within one season. We don't typically find those within later successional ecosystems, except on the edges or where there is disturbance. So that's kind of what we're talking about when it comes to weeds. So a lot of times we'll go on walks and we'll mostly find weeds along the paths. But when you get deep into the scrub or you get into your softball meadow masses, you don't have a lot of weeds going on there. You do have a lot of later successional grasses that might look like weeds, but they're actually perennials, and they'll go dormant and they'll come back. And those are a certain level of succession that need a certain level of fungi to bacterial ratio. So does that answer your question?
It does, because there's a definition of a weed.
Yeah.
I wasn't sure what you were.
Yeah.
I wasn't sure if we were talking about roses. And
Yeah.
another question.
Yeah.
If you have a good soil microbiome, and you live on land that has no soil microbiome.
Yeah.
If you took the top 2 inches of soil and transported it, would that be effective, or do you just need the leaf litter? How do you transfer a very healthy soil body to a site that you know has?
Yeah, so this is a great question too. The question for those of you that might have not heard it on the call, it's how do you inoculate a patch of land that might not have a lot of biology from one that's adjacent that does have biology? What's the best way to move some of that biology over? So this is where kind of ethical and responsible ways of collecting biology comes into question.
I own both.
Great.
So
Great. So yeah.
Yeah.
Yeah. So typically the best way is to, because what you could also be doing is moving pathogens at the same time. So I'll give you the ideal scenario. Ideal scenario is you collect a few, and what we're talking about are a few tablespoons. We're talking about very small amounts of samples, and it could be from under leaves, it could, dead leaves, it could be within, different levels within the soil, because there's different biology that lives there, and taking it from random places around and taking just small handfuls of basically of soil. The ideal situation is you make a thermal pile of compost and you inoculate that compost with those, because what that will do is it will eradicate any pathogens within the process. And then you take that compost because you have a larger volume now, and then you're able to inoculate your land with that material. What you could do alternately, if you don't have the capacity to make compost, is take some over time or all at once, take a few spoonfuls or handfuls of soil from different areas and inoculate them within the root zones of your different plants and just observe. Every system's a little bit different. It depends on the amount of moisture you have and what takes hold. It's kind of like, if you think of it, not all the organisms are going to survive as soon as you put them there. There has to be some sort of like pilgrimage that happens where, the first ones, half of them don't survive, and then, more come. You might have to do it more than once. You maybe do it, 3 times a year where you move certain amounts. But if you have fungi, if you're able to move fungi, and if you have insects within the system, you can trust that they're actually going to be able to move them around within the land for much faster than just, whatever the bacteria is able to move on its own. So ideally, you make a compost and you spread that compost via like an extract or a liquid, and then you actually inoculate the entire area with that. That is really the ideal situation. Yeah. Does that answer the question? I'm going to look at some of these questions here, just because we got a few that came in.
Eileen, I think was the first one.
Yeah, Eileen, she said, what causes sooty mold on plants? So this is one of the photos here. And this is something I brought up because when we're going on walks, oftentimes we'll see sooty mold on some of the shrubs that we're walking by. So why would they be there in a natural ecosystem? So I personally observe that they're usually there on the periphery along tracks and trails. And essentially, taking a step back, what is sooty mold? So sooty mold is a fungal infection, essentially, that happens when you have things like scale insects on plants. So scale insects will create, they're a sap-sucking insects. They'll attach to the plants, and they will take sap from the plants, and they'll produce something called honeydew that they excrete. Ants will actually harvest this honeydew, and oftentimes there's that symbiosis between the two organisms. But what also loves the honeydew is sooty mold. So it's the perfect sugary kind of environment for them. And you'll see a lot of plants will be very sticky and they'll look very shiny. And then a couple weeks later, you'll have a sooty mold infection. What this is telling you is that the plant surface is not covered in enough biology. The plant surface is denuded, and essentially there's nothing competing with the sooty mold once it lands. If your plant was actually covered in biology with the entire soil food web, there would be no chance for the sooty mold to take hold. And another thing here is also, if you have an infestation of scale insects, that is telling you that your plant is not getting enough nutrients to create denser cell walls, to create more complex lignins that it's able to protect itself. If a scale insect is overtaking a plant, that is telling you that the plant's immune system is compromised. So the question was, what causes sooty mold? And a lack of soil biology. That's what's causing sooty mold. So going back to when we're on trails, we have compaction via the trails and the tracks, via the maintenance. Sometimes there is spraying that happens, there's Roundup that happens along the edges to control some of the weeds, and that affects the soil food web. So a lot of these plants on the peripheries aren't the healthiest, and you also see it because that's where a lot of the invasives come in, and they're pioneers that don't need a lot of fungi. They thrive on bacteria. So that, I hope, answers that question.
So if a potted plant has sooty mold, what should I do?
So potted plants, and this is the thing here, I often get asked about potted plants. My plant is sick. What am I doing wrong? If your plant has yellowing leaves, if it's not an obvious thing like it's not getting enough water, if you're fertilizing it, what you think you're fertilizing it, and you're giving enough water and it's getting enough sunlight, but it's still very sick, the plant is not getting the nutrients it needs from the soil. And it typically means that you don't have a balanced soil food web. And the thing with potted plants is they're at our mercy. They don't have access to a larger soil area that they're able to have those interactions. So they're really dependent on what we give them. And you can either go the route and bypass the soil biology and give them, solubilized forms of fertilizer, and that's a lot of maintenance over time. And then you still risk burning the plants. You risk the plants not thriving over a long period of time, especially if they're complex plants like woody plants that need, a lot of different sorts of minerals, and you might not know exactly what mineral your plant needs. So why take the guesswork out of it? Put the soil food web in there, inoculate the plants with that, and have the plants and the organisms sort that out. They know exactly what they need. There's billions of years of evolution here. The plants are so intelligent. We all know this, and we all want to learn from them. So the benefit of the doubt of expressing their maximum genetic potential by putting them in The
I don't know where 10 is.
involved in
I
it.
can't put any of the others on the scale accurately.
Next question. Sue, my question is, understanding the simplified, is that native plants will grow where they best like the soil? If that is so, shouldn't the soil be understood before trying to introduce more native plants into an area to replace exotic plants? This is a great question. And we talk about this a lot because people come and say, I've tried this native plant over and over again and it doesn't work, it's dying. Okay, change your strategy. If the plant isn't liking it there, if the plant isn't working, it means it's not liking it there. It means it typically is a soil issue. So changing strategy, yes, you do need to know what's going on in the soil if you want to have the maximum success. And you can do it via trial and error, and you can plant all these different sorts of plants and see what sticks and what doesn't stick. But if you take a step back and you observe what is going on, what level of succession am I at? What level of succession do I want to be at? Do I want to pull my succession back because I have too many woody plants and I want to grow a meadow? You know, understanding where you want to be, yes. Knowing what's going on in the soil is really, really important. And this is where observing natural ecosystems comes into play. So, you know, what I do is I go out into natural ecosystems, I'll collect, you know, a teaspoon of soil, I'll take it home and I'll study what is going on under that microscope so that I can know, well, okay, I've looked at 20 samples of softball meadows and 20 different parks. They all have this fungi. These softball meadows probably need this fungi. So I need to make sure that if I'm growing these softball meadows, I need to be providing them those sorts of associations. Next question. Can you comment on the use of fish emulsions in the context of microbiology that happens in the soil food web? And how might this differ from how compost tea functions in the same environment? Does our South Florida native soil pose any unique challenges or opportunities in building a living soil? So lots of questions here. Fish emulsion is used oftentimes to feed biology, because what is fish emulsion? It's essentially bits of fish that are ground up together and mixed up together, and it has a lot of complex molecules within it. And fish emulsion sometimes is great to feed fungi because they're the only ones that are able to break down these different molecules within the fish emulsion. So we actually use fish emulsion to feed the biology when we're making something like a compost tea, where you've diluted the biology within a big tank of water, but they need food to replicate so that we can inoculate this entire tank of water with these organisms. So we add small bits of fish emulsion, which is basically the food for the organisms. And how might it differ from how compost tea functions in the same environment? They are related. Sometimes you can add fish emulsion diluted directly into the soil if you have the fungi, but if you don't have the fungi, that might go to waste. So you need to make sure that you have the fungi first before you add the fish emulsion, or at least know how much you're adding or why you're adding it. And does South Florida have our native soils pose any unique challenges? We have very shallow soils, typically very sandy soils, which don't hold a lot of water. So increasing your organic matter is so important, but not only that, increasing your biology will allow us to access a lot more minerals that our alkaline soils we don't typically have access to. So things like iron is a common deficiency in soils that don't have the right biology or that are depleted of organic matter. So there are specific challenges everywhere you go. And there's so much intelligence happening under the soil, and we are only just coming to understand how this happens. So we have a good understanding about the gist of how it happens, and we know a lot about the organisms, but we've only identified not even 1% of these organisms. There are so many different species that we're still learning about. So trust in what nature's doing. We want to replicate here in Florida Native Plant Society. We want to replicate native ecosystems. We want to trust in that intelligence. So inoculate the entire system of what it is. It's not just about plants. It's also about how they fit into a system. I'm rambling a lot here. Who else? So Sue asked a question. My question is understanding the, oh, no, wait, we already read this. Sorry. Richard asked, what brand of soil/fertilizer should we use moving forward? This is a big topic because I haven't found a source of biocomplete compost locally. I'm working on it and I'm studying how to make that. There are different people that make compost locally. And I'm in the process of going out and finding these people and testing their samples so that I can better understand what's missing or give them recommendations as to what they might want to be adding into those to make it a better product. And the best thing, if you can find, like I said, vermicompost, there's certain growers that provide vermicompost. That's a safe compost to use. It won't have a lot of fungi in it, but it's safe because you'll know that there aren't any pathogens in it. Because of the way the system works, there won't be pathogens, and that's the safest thing. I'll say honestly, I hope no one owns a big box store, but a lot of like plastic bagged compost that you get that's black when you open it, it has a weird smell sometimes. That's not compost, it's just organic matter. And if you look at it under the microscope, it doesn't lie, there's pathogens in there. So just be cautious and just observe, use your senses, smell, use your sight. If you see that it's black, you should know it should be 70% cocoa brown, and kind of go from there. And every neighborhood's different. Not all of us have access to every store. So we're figuring that out locally as to where we can get that stuff. What else? So Sue asked, where can we get the soil tested to match the soil for native seeding to make success? So this is what I'm working on. I'm about to do my last exam for my lab technician program, and you can call me and we can set up for me to take samples and look at it under the microscope for you. And there are other people elsewhere within the state, I think in the northern part of the state and elsewhere in the country. I think currently I might be wrong. There might be people in Miami, but I'm currently the only one in Broward that does microscope testing. So call me and we can figure something out. Diane asked, should we introduce some compost into the other areas of the garden now before winter or wait till spring? Winter and usually fall is a great time to inoculate your soils before dormancy happens. But also, I mean, Florida is a little bit of a different situation. I know Diane's actually in the Northeast, that's why I'm saying this. But also the spring is a great time because it also depends on how your plants are functioning. I think in the fall, your plants are, at least in the Northeast, they're moving a lot of those exudates into their roots. And a lot of those are going into the soil. So adding that biology and making sure it's in there during the fall is great to make sure that those organisms can grow during the winter as they're insulated under the ground. And then in the spring, inoculating your plant surfaces with that biology is great to make sure that they move into the season, into the summer season, and they're already covered with all this biology. You don't have to worry about any pest issues. Should I send a plane ticket to fix my fields before winter? We can arrange that. And then Anne finally asked, I have a yard packed with healthy natives. I've never fertilized and have really done nothing to make the soil healthy except leave the leaves. Great. I have lots of leaves breaking down everywhere. That's awesome. I actually once put a sign up to leave the leaves for wildlife, and I got called out by the realtor that was selling the house that it scared people and that they don't want to see that there's wildlife here. And it's funny, I was shocked that people are so disconnected to think, wildlife is something that's dangerous. If you're doing it wrong, yes, it's dangerous because it's anaerobic and you can be handling this material and you can get sick. But if you're doing it right and if you're observing how nature does things, it's perfectly safe. And it's actually really good for you because where does our gut biome come from? Naturally, it's from the soil. It's from the food that we eat that's covered in this biology, and some of them pass through and some of them survive. And understanding how we fit within the system, we're not disconnected from this. So yeah, leave the leaves. That's a great food for the organisms. And like I said, the best thing you can cover your soil with is living plants because they will mulch themselves. They will lose their leaves, they'll mulch themselves. You won't have to be putting so many inputs over time. So put a lot of input initially, do some sheet mulching, get that work in there, get that biology going, and then let it run, let it do its thing. And then
One last question.
Yeah.
Should I be using dyed mulch?
I actually have gotten asked this before. So it depends. There's different sorts of dyes that people use in mulch. A lot of times in mulch, you get preservatives that are added, so it lasts longer. I'm an advocate for using as natural a mulch as possible because you don't want the mulch to be there forever. you want it to decompose. You want it to feed the organisms to grow the plants to mulch themselves. So if you're using a dyed mulch, red mulch, which is a color theme that some of us might like or might not like. It depends. A lot of times it's colored with certain types of iron chelates to get that red color. Sometimes it's an artificial color. So understand what you're using, understand the product. If you have to use red mulch and that's all you have, make sure you have a really good compost that you're overwhelming the red mulch with, to make sure that anything that trickles out of that mulch that might be toxic, that you don't have a lab to send it to test to see if it's really toxic or not, just make sure you have the biology and just apply it. And then, maybe for 1 year you're using it, but then hopefully after that 1 year you have plants and you will never have to mulch again. So yeah. Any other questions? Yeah.
Is there any? Set around.
Yeah. Yeah. To a certain extent, yes. There are situations where you'll have pavers where you want to remove weeds from, the way I like to do it is I like to use boiling water, because it'll kill the plants instantly. You might have to do it 2-3 times depending on how vigorous the roots are. But it's the safest way because at least you might, hopefully that water cools off by the time it gets to the edge and ends up in your soil. There's a lot of unlearning that we all have to do, and I've had to do throughout this whole process of, what's a weed? Is that a problem? Is it okay to leave things in certain areas? I would say, mowing is not a bad thing in certain areas rather than eradicating the weeds with something that will like kill the biology, because vinegar will kill the biology. Vinegars are something that anaerobic organisms produce and they actually can burn plants, and that's why it works. I think in the short term, if it's a small area, it's not such a big deal if it's something like vinegar that decomposes pretty quickly in the environment. But then maybe try to come up with, more of a long-term solution. Why are there a lot of weeds happening there? Is there a lot of organic matter that builds up in that area? And do I need to redesign around it, to make sure that there aren't weeds? Or so the edge between turf and native beds. Yeah, so there's different ways to do that. I mean, sometimes we use physical barriers. We'll use like edging. We'll use like metal edging. We'll use like rocks that will create that barrier. And the thing with turf is it's something that's kind of become this cultural thing that we're all, it's functional. We use it. It's actually pretty useful sometimes in the yard to be able to have that open space. But there is kind of a level of maintenance that comes with it, one thing with that too is if you have turf that's going into a bed and kind of infiltrating a bed, it means that usually that ground is not covered enough to suppress the grass. Because if you have a very dense layer of plants on the edge that the grass is not able to, that overshadows the plants, that the grass that isn't able to penetrate that deeply, it'll weaken the grass. At least if it does infiltrate, it's not as strong as just kind of taking over. So maybe adding more layers in that planting bed to make sure that if the grass does infiltrate, it's not as intense, and then you can catch up with it and you can go in and do any manual labor that you need to keep it in check. I hope that answers the question. But I mean, ultimately what we want is to reduce the amount of turf, which we're working towards. Yeah. A physical barrier usually also, it can be a bit of an investment initially, but it does help a lot and also makes the maintenance easier. Yeah.
All of it is also, budget, like how you're someone want to commit to that, hiring outside. That's a whole balance that we all consider when we're thinking.
Yeah.
Investment, but long term, it's gonna make you feel far, you don't feel like you have to.
And long-term also understanding what is the, going back to succession, what is the fungal to bacterial ratio for the turf? And what is the fungal to bacterial ratio for the plant bed? If you're planting very woody plants, shrubs and trees and such in the plant bed, you're hopefully having a lot of fungi in that system. Those sorts of systems don't provide the nutrients necessary for that earlier succession. So if you have a lot of infiltration of grass in those beds, it's also telling you that you might not have, I don't know what sort of plants are in the plant bed, but you might not have, it's telling you that the fungal to bacterial ratio between the two systems is the same. If you have way more fungi in the plant bed, the grass will try to grow. It doesn't have enough nitrate, which it needs, depending on the type of grass, of course. And then it just won't survive. It'll actually try to grow, and then it just won't be as strong. And then it becomes easier for you to maintain that physically, over time.
But if you're in a low succession, like a meadow, and you have like meadow grass, low planting.
Then
right?
prioritizing what's important.
Much more important because then you're that long turf, long grass is looking at kids.
Yeah. Do we have any other questions online?
Someone had a question about the micro fungi. They wanted to scan.
Yeah, right. So you mentioned either that there are, earlier, that there was something that inhibits, blocks mycorrhizal fungi. I missed that, and can you expand? Sure. So in early successional systems, past weeds are typically things like, things like brassicas, kale and cole crops, mustards. Those require associations with actinobacteria. So actinobacteria is a type of bacteria, but it's filamenticious. So it'll look like a pencil thin kind of chain of bacteria under the microscope. And the way they process the nitrogen and the nutrients within the soil is really conducive to things like mustards and kales and brassicas. But they also have a chemical inhibition to mycorrhizal fungi, which are essential for anything above that succession level. Anything that's late successional grasses, vines, trees, they need, many of those plants need mycorrhizae. So the actinobacteria actually act against the mycorrhizae, and they inhibit them from spreading and being able to take hold in the soil. So understanding where you want to be, if you're growing a vegetable garden and you're trying to grow kales and broccolis and cabbages, you want actinobacteria, you don't want mycorrhizae. So you want an early successional system so those plants have the nutrients available to them easily without you having to put that much work into it. So I hope that answers that question. And then steam machine to remove lawns or large amounts of weeds. I mean, that's like adding boiling water, to kind of desiccate the plants. That could be something where you're maybe on the surface killing things, but then not so much hurting things because it's going into the soil. One thing, I don't want us to spend too much time, but I do want us to look at these slides because this is pretty useful. As some of you are going to go out now and you might be like seeing these. So these three images look very similar, but they're quite different. So the one on the left is what we want to see. This is beneficial fungi. And the reason we know it's beneficial is because it's creating kind of a network pattern. If you look at this under the microscope, those fungal strands that I showed you in that previous view, they'll bind together and they'll create these complex kind of networking systems that reach out and branch out into the soil. It can look something like, if you're grabbing a bunch of like loose soil, it'll look like that. And you can see clear strands. Sometimes they're white, sometimes they're tan. They can be all different sorts of colors, even brown and red sometimes, depending on the species. So that's actually good. You want to see that. And that means that, if you see mushrooms and you go digging, you'll probably see something like this. And this is really great. What you see in the middle here, if you have a compost pile and you see this, it's more like fuzzy wuzzy. It's not really, it's kind of amorphous. It's kind of like a mold. That's actually harmful fungi. So a lot of mold, like penicillin, it'll make, it has antimicrobial effects. So it can actually detriment the bacteria very, very badly within the soil, good bacteria that you need. So if you see this, it means that the system is going anaerobic and you need to provide more oxygen into the system. So if you see this within your pile, if you open up a compost pile and you see mold, open up the pile, let oxygen run through, and then also make sure that you inoculate the pile with good organisms that will eat this when it's still in a microscopic form before it gets out of hand and you can actually see it with your eyes. Because I think this is like millions and millions of strands that are actually so large that you're able to see them with your eyes. And the one on the left, on the right here, oftentimes if you'll get wood chip piles and you let it sit there for a day or two, you'll go to open it and you'll see this kind of white cast within the pile. That isn't actually fungi. So that's actinobacteria. That is the actinobacteria we were just talking about, the strands. So what's happening here is we talked about aerobic organisms. We talked about anaerobic organisms. We have organisms that live in between, that live in both conditions. And actinobacteria live kind of on the brink between those two scenarios. They can't compete with organisms when it's fully aerobic because of how they metabolize within that oxygen level. And they also can't compete with fully anaerobic organisms. So if you see this and it looks like a white powder, it means that your pile, the center of the pile, is going anaerobic. It's going anaerobic. It's tending towards an anaerobic condition. And what happens here is there's so much food from this organic matter that the actinobacteria are eating up all this food and growing at such an exponential rate. And think of how bacteria grow. You can have one bacterium that will split into two within, it varies, but within 20 minutes. So if you do the math, within 24 hours, you have 1 million bacteria from one bacterium. So this is what's happening here. They have all this food. They're growing exponentially in numbers, and they're also using up all the oxygen within the center of the pile. Not enough oxygen is able to move into the pile fast enough. So it's going anaerobic. So the actinobacteria are thriving because it's a lower oxygen condition. They're not competing with these oxygen-rich organisms. And this tells you, okay, I got to open up the pile. I got to get oxygen in there so that I can make sure that my good organisms aren't dying or going dormant. So as soon as you see this, open up the pile, spread the material as much as you can, water it down, get the water to allow some oxygen to pass through there, and you'll avoid seeing that. But if you do want to grow mustards, kales, brassicas, you can take some of this and add it into your soil, and it actually might be really good for them. So it really depends what kind of level of succession you want to be at. So this kind of concludes. Thank you all. This is some footage. This isn't my footage, but this is a great YouTube channel if anyone wants to get into this. And it shows what some of the things under the microscope will look like. So this is actually a rotifer. I'm going to close this so we can see. This is a rotifer. This is another facultative anaerobe that lives kind of on the cusp between aerobic and anaerobic. So if you see this within your pile or within your sample, you know that it's going anaerobic. That's a very clear sign. So there's a lot going on here. Things are zooming by. I mean, this is what I see under the microscope. I mean, we weren't able to do a sample today, but hopefully if you guys are interested, we can do another presentation another time. We can get samples in and we can bring a microscope and actually look at these. These are ciliates. We call them race cars of the microscopic. They zoom by. A lot of these samples are taken from ponds, which are anaerobic, and that's a different system. And so these are anaerobic organisms. They're ciliates. They process nutrients in a different way within the soil. And like I said, yeah, it's really fun. I mean, it's like a safari ride, like looking through, and you can sit there for hours looking at these things. So these are stentors. These are actually unicellular. They're one cell, and you can even see them with the naked eye. They can grow as large as like a millimeter, and you can actually see them within. They are anaerobic, and you can see them within water samples. Stalk ciliates here. You can see how they're filtering bacteria as they move along. I mean, this is happening under our feet, everywhere. We don't even see it, and it's dictating how natural ecosystems function. And it's really, to me, I find it just so mind blowing every time I look under the microscope. It's really, really nice. So thank you all so much. It was a pleasure to present all this information. I hope I didn't talk your ears off. And everyone online, thank you for tuning in. if you have any more questions that you weren't able to ask, maybe just email Broward.
Yeah, you can
FNPS.
email, yeah, Broward chapter fnps@gmail.com, and I can forward them on to the board
Yeah.
directly. Yeah. Thank you all. Thank you all for
Thank
coming.
you so
We appreciate
much.
it. Y'all, thank you.
Thanks, everybody.
We're gonna linger for a bit.
We gotta clean up.
If anybody's interested, I can bring some.
There's a way.
It's good